All right. So for the single opponent cell, let's just stop there for a minute. Do people have questions on that? Does that make sense to folks how that would work? Yeah.
So if the center responds best to long wavelengths, do we have, is it responding to a range of wavelengths or just one specific wavelength and we have multiple cells for each?
No, so yeah, so this would be, I mean, what we're really talking about here is like, what we're really kind of saying is that the long cones, or the quote-unquote red cones, are the ones that kind of feed into the center of this cell's receptive field. So in other words, like, the more those red cones are stimulated in the center of the cell's receptive field, the more this cell would respond, right? So that, yeah, so it's a little more, I don't know if that's more or less complicated to think about, but in other words, like, you know, presumably if the center of the, so like lots of different wavelengths stimulate red cones, but the things that stimulate red cones the best are, you know, kind of that ideal red wavelength, right? So the best thing would be the optimal red wavelength, but lots of other wavelengths would stimulate this some, right? And similarly, you know, the thing that inhibits the surround most would be that greenish wavelength that is like the most preferred wavelength by the green cones, or the medium wavelength cones, but actually lots of different wavelengths would kind of stimulate those medium cones and thus, you know, inhibit the cell's response if it falls in the surround. So technically speaking, yeah, like, because, so, you know, I fudged a little bit by saying like, well, you could have red light. Technically, red light will also stimulate a medium cone to a certain extent. So actually, better still than this would actually be to have red in the center and total blackness in the surround. But that's a sort of fine detail we don't really need to worry about if the basic idea makes sense. Other questions about this before we move on? Okay. So what this gives us so far, these single opponent cells, is, you know, this gives us cells in your nervous system that are basically detectors for spots of one color, right, that are not surrounded by other colors, or, you know, that are not surrounded by particular colors, right? So you have something that likes red in the center, doesn't like green in the surround, responds pretty well to red everywhere, or to red surrounded by just, you know, darkness, whatever, right? But it does not like green for sure. Actually, it doesn't really care if it gets green in the center. I mean, that doesn't hurt it, it just doesn't help it because it wants red light in the center. But it definitely doesn't like green in the surround. Okay, so if that makes sense to you, now then a later stage of processing would have something that looks a little bit, in terms of what the ideal stimulus is, it looks a little bit more like what we're used to seeing for pure brightness. And this is called a double opponent cell, because it has a kind of center surround opponency like the cells that we're used to that are achromatic, that just prefer light in the center, dark in the surround, or vice versa. But it's more complicated because you have kind of like two colors, or two like color pathways that kind of have to be involved to make this happen, right? So essentially what this is saying is we want a cell that, kind of like retinal ganglion cells for the achromatic stuff, you know, prefer a contrast in the center versus the surround, right? That like that basically, this is going to be a cell that prefers a redder center and a greener surround, right? So it wants the center to be redder than the surround, and it wants the surround to be greener than the center, right? So that is like those other receptive fields we saw that like spotted light surrounded by dark, or spotted dark surrounded by light, but now it's kind of color specific. But in order to do that, basically what you need essentially is a center that is excited by long wavelengths, or red wavelengths, and inhibited by medium wavelengths, or greenish wavelengths, right? So it's more complicated than the sort of properties of this cell, which is basically kind of a detector for a dot of light of a particular color. This is now something that likes a, you know, a kind of contrast of the center versus the surround, but for that to happen, you need to not only be excited by red in the center, like the single opponent cell, but also inhibited by the green in the center, right? Which was not true of the single opponent cell. The single opponent cell didn't really care how much green light fell in the center, right? And similarly, the surround not only dislikes a particular wavelength of light, which in this case would be long wavelengths, right? It not only dislikes red in the surround, it also likes green in the surround, right? Which is, again, kind of different from up here where it just liked one thing in the center, it disliked one thing in the surround. These double opponent cells like and dislike, you know, two different things in both the center and the surround, and what they like and what they dislike in the center versus the surround, you know, is switched, right? So it likes long or red in the center, dislikes green in the center, or medium wavelengths. Likes green wavelengths in the surround, doesn't like red in the surround, right? So if you look at what the cell would respond to, it's response properties, if the cell's receptive field fell on these same four positions in this kind of red-green field, would be different, right? So before, this was the worst stimulus for this guy, right? But this kind of fully red, or actually this fully green or this fully red position in the receptive field, those would be kind of like medium or neutral responses, right? Because this cell just kind of like, hates green everywhere and loves red. Well, hates green in the surround and loves red in the center. So this cell isn't getting anything it likes, and it's getting everything it likes over here. This cell, it likes red in the center, dislikes green. So that's not good, but it likes green in the surround and dislikes red, and that part's good, right? So those two kind of cancel each other out. Conversely, over here, this receptive field for the same cell would be getting what it likes in the center now, but it's not getting what it likes in the surround, because it doesn't like red wavelengths or long wavelengths in the surround. So again, those kind of cancel each other out, right? So for this particular cell, the double opponent receptive field, its worst stimulus would be basically this. I mean, this is not the worst stimulus possible, but it's the worst possible on this screen. So the worst thing for it, right, would be a spot of green surrounded by red, right? So, it doesn't like the green in the center because it doesn't like medium wavelengths in the center. It also does not like, sorry, I'm going to screw this up.